In this video, we will discuss how the glycosylation of the Flavivirus envelope protein helps in the cell attachment, or we can say the first step for the virus to infect the cells. So these are the different variants of the Flavivirus uh, envelope protein sequence is mentioned, and in this we are able to see that uh, at two position the N glycosylation sites are available. First one is the second domain of the envelope protein, that is at 67 amino acid sequence from N site, and another one is 154 amino acid sequence when it is read from N site. This is the standard from where we start to read the amino acid, and we can see that the asparagine linked or glycosylation sites at 67 is conserved only in the dengue virus, whereas uh, the glycosylation site at 154 is conserved in most of the other variant of flavivirus also. And this flavivirus is usually a uh, positive strand single stranded RNA. Its genetic material is single stranded RNA, and it also has the envelope protein as it is understandable. So what people have done is people have tested whether this glycosylation sites or the glycosylation at these sites will help in the cell, will help the virus or flavivirus to attach to the cell surface or not. So they did two kind of experiment. First one is that is that they uh, first experiment is that they test this flavivirus without any glycan sites, or by removing these glycan sites through some enzyme treatment such as silidase treatment. So in that case they have observed that the flavivirus attachment to the DC sign lectin and DC sign R lectin is reduced, or they did not see any attachment, or we can say that minimal binding is there in case there is no glycan. However, when they keep the glycan at N61, they did not disturb. They use the wild type flavivirus cells which has the glycans at N67 or glycans at 154. They can see the attachment of the cell surface. They can see the attachment of this virus with this DC sign receptors and also DC sign NR receptors. And when they did another customized study in which they test the different kind of glycans, and they find out that the complex glycan at N67 position attached to the DC sign, whereas the complex glycan at 154 is attached to the DC sign NR. These signs are more preferred, and this complex glycan attachment to this DC sign is somewhat observed, but they are not that significant compared to the this high mannose glycans to the DC sign. So DC sign NR can attach both the complex and the high mannose glycan, whereas DC sign can attach helps in uh, or DC sign can helps in attachment of envelope protein that contains more of this high mannose glycans. So this experiment substantiates the point that glycosylation in the envelope proteins helps in the virus attachment to the cell surface, or we can say the first step for virus infection. So by targeting the glycosylation of the viruses and at the envelope by targeting the envelope protein glycosylation of the viruses can be one of the useful strategy towards uh, making those virus uh, non-efficient or less efficient. And this is one of the strategies that people are targeting, as the glycans, apart from helping the virus um, in the virus infection by playing playing a significant important role in cell surface, by playing an important role in cell surface attachment of the virus to the host cells, or we can say attachment of the virus to the host cell surface. It also has this glycosylation also helps in shielding the envelope protein, so that uh, the immune the immune system of the host cells will not be able to recognize, or find difficulty in recognizing the uh, envelope protein as a harmful substance, or find it difficult in the identifying the virus as a harmful substance. So these are the two main roles of glycosylation on the envelope protein of the viruses. I hope you have understood the importance of glycosylation on the virus and how they help in the virus infection. If you need to understand these things in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.